It's with great pleasure to introduce our speaker today, Dr. Susan Harris, in celebration of her 25 years at Tufts. Susan is a scientist one in the bone metabolism lab, and she's also an associate professor at the Friedman School. She's actually one of our own Friedman grads. She did her master's degree at the Friedman School in Nutrition, and then she did her doctorate training in epidemiology at Boston University's School of Public Health. While most of her career has been here at the HNRCA, we did lose her for a few years when she went to the New England Research Institute as a senior research scientist in their Institute for Community Health Studies. However, we got her back, and we're very grateful she came back. She's been an incredibly productive scientist. She has over 70 original research publications, and her area of expertise, of course, is vitamin D and other nutritional determinants of chronic disease. So without further ado, I'd like to introduce Susan, who is going to talk to us today about vitamin D answers and questions. Thank you.
Thank you, Sarah. So it's actually closer to 30 years ago that I came to Tufts, which is when I started my master's degree process. And back then, this was the School of Nutrition. There was another building just like it next door, but this was it. It wasn't the Friedman School. It was just the plain old School of Nutrition, Science, and Policy, and it was here. And this is where I had the good fortune to meet Jerry Dalal, who's hiding way back there in the corner. And I took his statistics class, and that class really developed my interest in data analysis, which to this day remains my favorite part of research. And knowing Jerry was useful in other ways as well, because through having been his teaching assistant, he got me all of my subsequent jobs, really. The first one was right here in this building, in this, that was my window. I looked out on a tree and I analyzed African household food data. A little while after that, I went to Harvard at the Institute for International Development, and I analyzed other African food data there. Jerry got me both of those jobs, kind of indirectly on the second one. And the Harvard job was just running out. The project was running out of money, and the phone rang, and I was in my cubicle working in D.Base on a computer like that big. And the voice said, "Hello, this is Bess Dawson Hughes. I'm at Tufts University, and I got your name from Jerry Dalal." So I came over here and I talked to Bess, and with a couple of years off, I've been here ever since. So I've actually worked in many areas other than vitamin D, but vitamin D is one thread that's continued through my entire time here at the center. So today I'm going to focus on vitamin D, a lot of our work, but I'm also throwing in a little bit of other people's stuff just to kind of get across some of my thinking about where the field is going and what we need to be thinking about in the future in vitamin D. When I first arrived fresh with my master's degree, I walked into a series of clinical trials that were going on in our laboratory. And any of you who has ever worked on a clinical trial knows how hard these are. I put the study numbers on these because both so you can see how low they are, but also because any of you from the NEL or the MRU, you knew these studies by their numbers, and you probably haven't forgotten the tube after tube after tube after tube that you spun, or the volunteer after volunteer that you saw for these studies. This was the first trial that began while I was here, and it was first of a series of vitamin D trials. In this one, we had postmenopausal women. Now, you know that vitamin D varies with season because the quality of sun exposure varies with season. So this adds an extra logistical difficulty to any studies of vitamin D because you have to consider season when you're designing the study. So the way we handled season in this particular study was we enrolled everyone in June or July, Well, imagine that. I mean, this wasn't just 1 year. I think we enrolled over 2 years, but trying to recruit all of these people and enroll them and get their baseline measurements in 2 months out of the entire year. Very, very difficult. And then we measured them every 6 months. And then they were randomized in this study to 500 units of D or placebo with usual intakes of 100 units of D. In all of these slides, I'm going to try to get across what the total intake was, because I think that's one place we go wrong in making generalizations about the findings from vitamin D studies, is not taking into account where people were before they began supplementation. So these women were right around 600 units per day, which is the current RDA for adults under 70. We did more than one bone site, but I'm just showing you here the results at the lumbar spine. I found the findings from this study really, really interesting, not because of the overall effect of vitamin D, which of course we were happy to see, but because of the seasonality in the changes in bone density. So we have 2 periods here. I knew I would do that. June, July, which you can roughly think of as summer because it contains the peak levels of vitamin D, and the second one, which you can roughly think of as winter. And in both the groups, placebo and the vitamin D group, they gained bone mineral density during the summer significantly. In the winter, both groups lost bone mineral density from the spine. But those who were receiving the supplemental vitamin D lost less bone mineral density than those on the placebo, and this led to a net overall benefit over the period of this 1-year study in bone mineral density. So on to the next study, where we decided to compare 2 different doses of vitamin D, 700 units versus 100 units, again, postmenopausal women, and this same arduous schedule of enrolling in June or July. And again, the same usual vitamin D intake of 100 units. In fact, it was many of the same volunteers. And again, I've only selected one skeletal site here, but you can see over this study, which was longer, it was 2 years, that we have this seasonal fluctuation in bone density again. But you can see that over the 2-year period, there was a significant difference. Oh, I keep doing that. That button's in the wrong place. There's a significant difference between bone density at the end of the 2-year study between the supplemented and the not supplemented. I should point out also, though, that most of this difference between the groups had already occurred by the end of the first year, and these two slopes here are roughly parallel for the rest of the study. And this is something that we see repeatedly, that the maximum benefit from increasing calcium and or vitamin D is early on. And then our last big trial of vitamin D was the Stop It study, which was known by more than Study 620. And this was a multi-site study, ours being just one of them. But these findings are just from our site. And here we have somewhat older adults. They're 65 years and older, and we added for the first time men. So we have both men and women. We gave 700 units of vitamin D, so we've gone up a little bit on the vitamin D. We also added in 500 milligrams of calcium, which was enough to bring them up to around the RDA for calcium. We had 389 completers in the study. Their usual vitamin D intake was around 190 units per day. We did see some differences in bone change over the 3 years, but what we were, I don't know, I won't say we were surprised, but we were pleased to find that vitamin D also seemed to reduce fracture rates in this group. Now, this is a pretty small study to be studying fracture rates. Nevertheless, you can see that over the 3 years, the cumulative incidence of first non-vertebral fracture over those 3-year periods was about half that in the group that received the calcium and vitamin D compared with the group that received the placebo. Now, we felt that this difference in the fracture rates was too great to be explained simply by the reduction in bone density. The bone reduction in bone density wasn't big enough to explain that. So this study kind of led us to our thinking about other ways that vitamin D might be affecting fracture rates, perhaps through effects on the muscular system, on falls, on other things related to fracture. And many of those areas we've gone on to explore, although I don't really have slides on most of that today. Around about this time, I started to get interested in blacks and whites. It was already known from cross-sectional studies that 25-hydroxyvitamin D levels in black adults tended to be lower than those in white adults. I'm sure all of you are familiar with 25-hydroxyvitamin D, but just in case, it's the metabolite of vitamin D that's thought of as the storage form of the vitamin. It's the one that best describes vitamin D status. So this study number 770 was an observational study, and we recruited 90 younger women, this time aged 20 to 40, and we measured blood serially. So there had been, and there continue to be, many studies of seasonal differences in vitamin D, but there are very few that take the same group of people and measure them repeatedly through the year. So this one was interesting to me at the time, and I still like this slide. And you can see a few things from this slide. You can see that how many times am I going to do this? At every time of year, the white women have substantially higher 25-hydroxyvitamin D than the black women, substantially higher. You see that in both groups, there's seasonal variation in 25-hydroxyvitamin D, with the highest levels in June, July, and the lowest levels in February, March, which is what we routinely see at this latitude in Boston, 42 degrees. And finally, you can see that the amplitude of the change in 25-hydroxyvitamin D is greater in the whites. So in the summer, they're going up more than the black women are. And to me, this is an argument that at least some of the black-white difference is in fact due to the effect of skin pigmentation. I mean, obviously it is. Skin pigmentation has been shown in experimental studies to block vitamin D production, but as you'll see a slide later on, there's some thought that there may be other things that contribute to the black-white difference in 25D levels. And I did another study. I did this in collaboration with a physician at the Kearney Hospital. This was another observational study, and we went into low-income housing units in the Boston area. I can't remember how many there were, maybe 5 or so. And we invited all of the residents to come downstairs and get their blood drawn and have a heel bone density measurement and answer some questionnaires. All of these individuals were 65 plus. They were mobile enough to make it downstairs, but they were, there was some frailty among them. We measured in winter. Winter was a very, very busy time in our lab all the way around. And you can see here, although it's cross-sectional, the same thing that we saw in the younger women, and that if you look at the two lines with the white shapes on them, those are the white male and females, and males and females, and the ones with the black shapes on them are the black males and females. There wasn't much, if any, difference between the sexes, but you can see that the two curves for the white men and women are shifted to the right, showing that they have overall higher 25-hydroxyvitamin D levels. Now, on all of these slides, our assay methods evolved over time, as did those of others, so you can't take the absolute numbers for 25-hydroxyvitamin D too literally in comparing one slide to the next. This particular assay reads higher than what we're using now. So what we were finding with vitamin D levels was in keeping with national statistics. So this is a slide from Ann Looker, and you can see in the middle bar that substantially more of non-Hispanic blacks of all ages had 25 hydroxyvitamin D levels less than 30 nanomoles per liter, which they're using here as an indicator of deficiency, and less than 50 nanomoles per liter, which they're calling inadequacy. There's been, as you probably are aware, there's been a lot of discussion back and forth for the terminology and the cutoff points for what is deficiency, what is inadequacy or insufficiency. So we also measured bone density in the younger women, and this was just beginning to become apparent at that time that despite these low 25-hydroxyvitamin D levels, that black adults, and later we know black children, have quite a bit higher bone mineral density. So in this particular group, this is total body, but it was the same thing at the other sites. The total body bone mineral density was about 7% higher in the black women compared to the young white women, and when you adjust for various things, that difference was reduced to a little over 5%. We adjusted for all the things that mattered, and it's, I think, of interest to me anyway, that many of the things we adjusted for, BMI, weight at age 12, age at menarche, number of births, amenorrhea history, southern residence, which in many cases included birth in the Caribbean, and alcohol consumption, except for alcohol consumption, these are things that go back in time to early childhood. So this is a cross-sectional study, but we're by adjusting for things that preceded current bone density by many years, we're accounting for some of the difference between blacks and whites, a little bit, not much. And we now know that differences in bone density occur between blacks and whites very, very early in life. And some of this is undoubtedly genetic. We have explored a few genotypes and looked at this question, but this was a long time ago. This was in 1997. We didn't have the elegant genotyping methods that we have now. But we had a group of 154 young women. It was those same young women with some others added. And we looked at one particular polymorphism related to the vitamin D receptor. And in this case, and again, these numbers are small, but you can see that across the genotypes going from big F to little f, that there are declines in bone density of the femoral neck in the white women, but we don't see that in the black women. And perhaps more interesting is that the distribution across these genotypes is different in the blacks and the whites, so that 65% of the black women were in the favorable big F genotype, compared with only 37% of the white women. Now, this is, of course, nothing definitive, but it's just to give you a taste of one of many genes related to vitamin D that appear to differ between blacks and whites. Another one that we looked at that I don't have a slide on related to vitamin D binding protein. Now we're hoping to collaborate with the genomics group to look at some larger samples, not necessarily at blacks, but at some of the vitamin D relationships with genotype. So going back now to the older blacks and whites, so they have, and they also have higher bone density, which I'm not showing, but so we know blacks have higher bone density, lower 25-hydroxyvitamin D. Why? And this would be interesting to know. And is it because somehow they have a different relationship between parathyroid hormone and 25-hydroxyvitamin D? So we looked at this possibility. This is, again, cross-sectional. And you can see here that the slope of 25D relative to PTH is exactly the same in the blacks and whites, suggesting that no, the answer is that they have the same relationship between 25D and PTH. Now you'll also see that the PTH is higher at every level of 25D in the blacks for unknown reasons. I mean, I have some hypotheses, but that's not because of the 25D, so that was not an answer. And so we thought, well, maybe it's this, having this elevated, oh, let me go back a sec. You can see that green line there in the middle. The people with the points above that have hyperparathyroidism, so elevated PTH due to their low vitamin D levels. So we said, well, maybe bone turnover differs according to PTH status. So bone a higher bone turnover is a bad thing in older adults because bone formation tends not to keep up with bone resorption. So osteocalcin is a marker of bone formation. Serum NTX is a marker of bone resorption. And you see here that the black bars are those with the elevated PTH, and that in the blacks, as in the whites, and this doesn't achieve statistical significance everywhere, but in both groups, those with the elevated PTH have higher bone turnover. So this would suggest that vitamin D deficiency, secondary hyperparathyroidism have bad effects on bone in blacks as well as whites. So that was my going hypothesis up through 2001. And I was interested in doing a trial of vitamin D and bone loss in blacks and whites, but I never did that trial, but Aloya did, and this is a really fascinating study. One of the few I'm bringing in from others because it just says a lot about the things that I have thought about over these 25 years. So they looked at other skeletal sites as well, but I'm focusing here on total body bone loss. And this was a 3-year study. They had 208 black women with mean age 60 years. I believe they were all postmenopausal. They gave everyone calcium intake to bring them up to levels of 1,200 to 1,500. The dose that they were randomized to initially was 800 units per day or placebo. And for the first 2 years of the study, that's what they took. But then after the second year of the study, they revised the protocol and they increased the supplement intake to 2,000 units per day or placebo. If you look at the chart, the graph, the table on the left, you can see that both groups started out similar at baseline with 25D levels around 43, 47. So that's a little inadequate, not horribly deficient though. And by the end of the 3 years, with that changing dose, the supplemented group got up to 71 nanomoles per liter, which is very close to what many of us think should be the target until we learn more of 75 nanomoles per liter. So now you look at the graph itself and you can see that there was absolutely no difference in bone change between the groups. Absolutely none. I don't even have a indicator on here which are the blacks and which are the whites, and I noticed that at the last minute. I thought, oh, I better put that on, and then I decided it really doesn't matter. They're exactly the same. So they found no effect of vitamin D supplementation on bone change in this group of black women over 3 years. So a paper came out recently, just I think it was the end of last year, about vitamin D binding protein in blacks and whites. And this was a really interesting one as well. These were community-dwelling people, wide age range, 30 to 64, living in Baltimore, a little bit south of here, a little more effective sun exposure. Very large group, 2,085 men and women. Right? And they measured 25-hydroxyvitamin D in blacks and whites, and you can see in this slide that the distribution for the whites, which is the lower pink mound, is shifted to the right, suggesting that in this group, as in virtually every other group, 25-hydroxyvitamin D was higher in the whites than in the blacks. But they also observed that vitamin D binding protein was 50% lower in the blacks. And to my knowledge, this is the first time that vitamin D binding protein has been measured in a really large group like that. And what they decided to do was to adjust these original total 25-hydroxyvitamin D measures for vitamin D binding protein, and they had their own equation for doing this. And what they came up with, they decided to call bioavailable 25-hydroxyvitamin D. So this kind of follows the model of testosterone, where there's testosterone, there's total testosterone, there's free testosterone, and we think of free testosterone as the active part of it. And when they calculated these bioavailable values, they came out with distributions that were right on top of each other and concluded that 25-hydroxyvitamin D actually did not differ, rather that the bioavailable 25-hydroxyvitamin D did not differ by race. Well, this paper got hit by letters. I think there were 5 letters in the first issue that followed it, very, very critical on both methodological terms and physiological terms. The methodology had to do with different genotypes not being read in the same way by the assay. The physiological argument had to do with arguing that really it was bound vitamin D that should be considered the bioavailable portion because of the way that it was taken up into cells. So it's where we'll go in this argument, I'm very interested to find out. But I think regardless of whether this so-called bioavailable vitamin D is accepted, I do think that this paper really provides a reason for us to start paying attention to vitamin D binding protein as it affects the difference between blacks and whites, but in general in the vitamin D work that we do. Okay. So after the Aloya paper and after in general we had kind of felt that we'd done our thing with vitamin D in bone in a way, and for me personally anyway, I decided to take a look at vitamin D as it related to other things, mainly diabetes. And especially because I was still interested in blacks, and I thought, well, why not pick a condition that's more prevalent in blacks rather than less prevalent in blacks to look at a relationship with vitamin D. So over this time, between a little bit after I started and now, the number of people getting involved in vitamin D research has just skyrocketed. I mean, probably this is true for every field, but in 2013 alone, there's like 2,700 vitamin D papers. It's just has become impossible to have a global understanding of everything that's been published about vitamin D and to synthesize it and to really understand what's going on. And so picking the targets and what you want to work on has become more challenging. And I had to laugh at this paper in the Lancet, and there's another paper that came out more recently that's quite similar, because it's a systematic review of meta-analyses. So all in all, what's discussed here involves 62,000 individual people. So there's all these individual people, and then they get put into 14 different studies, and then the 14 studies get put into one systematic, the 14 studies get put into this meta-analysis, and then it was reviewed here along with a lot of other things. I'll show one more slide. But I'm going to make several points about this slide. One is that nothing against meta-analyses, but when you see the results of meta-analyses, you're very far removed from the individuals and the differences among them that may account for the findings to some degree. So what were the findings in this study? Well, this particular slide is from prospective cohort studies. And in this study, they had, it was a large group, a lot of deaths. They didn't actually report the follow-up period in the paper. You had to go find some supplement, which I didn't do. But they saw a substantial reduction in mortality. So this isn't necessarily, cardiovascular disease or diabetes, but in overall mortality in the first quartile of 25-hydroxyvitamin D compared with the fourth quartile, the relative risk was 0.71. So that's like more than a 30% reduction in mortality based on your 25-hydroxyvitamin D. So if this were true and this were causal, if vitamin D were actually reducing mortality to this degree, you would clearly want to be in the fourth quartile, or at least I would, in the absence of any other knowledge. So, this is basic. We know that, observational studies, we can't always know. We can't prove causality. One possibility is that vitamin D deficiency in these cohort studies is causing disease, and the other possibility is that it isn't. Okay, seems like a silly slide. I'm going to add one more line to that later on. Then this same paper by Autier goes on to look at the randomized controlled trials, and when you look how many of them, and they had some quality criteria and some size criteria, and so this isn't all the clinical trials by any means, and yet you can see that there are quite a few of them that included mortality as an endpoint. And here, I don't want to spend too long on this, but you can see that the relative risk for mortality in those who received a vitamin D supplement, and the doses are different, are varying. The relative risk ranged from 0.93 to 0.95 for the ones that were significant. So this is a much smaller apparent benefit of vitamin D. And I just picked this one paper, but this is something that we're seeing over and over with vitamin D, is that we're seeing these strong associations from observational studies and weak or null findings from intervention studies. And what do we make of that? Now, in my view, even a 4 or 5% reduction in mortality might be enough for me to pay attention to my vitamin D status, but by most standards, this is a pretty small effect. So the nice thing about this center and being able to do these elegant, smaller scale studies is that they give you the ability to really drill down and know who you're looking at. In this study, this was led by Dr. Tasso Pittas over at the medical school, and he used our stop-it data to do it. It was a secondary analysis. And so you may recall from the earlier slide that these were men and women aged 65 and older, and they were given 700 units of vitamin D3 and 500 milligrams of calcium compared with placebo for 3 years. And Tasso went on to divide the group into their status at baseline. He excluded people who were diabetic at baseline, and he divided the remaining people into those with normal glucose tolerance, which are on the left-hand side of each panel, and those with impaired fasting glucose at baseline. We only had fasting measures in this study. And then he looked at the 3-year changes in glucose and the 3-year changes in HOMA-IR, which is an index of insulin resistance. And he found, or we found, that among those with normal glucose tolerance, there was no difference. The supplement had no effect on the change in their glucose over 3 years. Whereas in those who started out already impaired, the vitamin D supplement led to smaller increases in glucose over the 3 years. So you can think of it as slowing down the progression of increasing glucose in the direction of diabetes. He found exactly the same thing with insulin resistance, so that those with normal HOMA-IR at baseline, there was no effect of supplementation. Those who were supplemented had a better change in, less of an increase in insulin resistance over the subsequent 3 years. So I don't know if Tasso's study was done at that time or not, but I wanted to look at something similar in African Americans. So we already had the idea by this point that the people we want to look at and what we're fortunate enough at a place like this to be able to get are those who are at risk in some way for whatever it is that you're trying to prevent or trying to prevent. So this was a small clinical trial. We enrolled 100 African American men and women. They were age 40 and older. 89 of them completed the study. They had to be overweight or obese, and some of our recruitment team is here, and we found that if we found African Americans who were overweight or obese in this age range, that they were almost always pre-diabetic. So that was our screening. We didn't lose as many people at screening as we thought we might. So that was bad for them, but helpful for us. And we randomized them to 4,000 units of vitamin D3 or placebo, and we followed them for 3 months. They had oral glucose tolerance tests at baseline 6 months and, baseline 6 weeks and 12 weeks. Why do we give them such a high dose of vitamin D? We already knew from preliminary data that it takes that much to get them up to the level we were looking for, which was around 75 nanomoles per liter for most of the subjects. So at baseline, the groups were pretty well balanced in terms of percent female, age, BMI, 25-hydroxyvitamin D, which was 38, 39, so much lower than what we were aiming for, which was 75 nanomoles. And they were similar in their fasting and challenged glucose levels. This was funded by the American Diabetes Association. So we were very successful with this supplement dose in getting the subjects to where we wanted them blood-wise on the 25-hydroxyvitamin D. They got up to, the supplemented group got up to 81 nanomoles per liter, so, and most of them, not quite all, but most of them got to 75. Now in the graph on the left-hand side, you can see the percent change in 25-hydroxyvitamin D in the two groups. And in the next two sets of bars, you can see the effect on fasting plasma glucose and 2-hour challenged glucose. There was absolutely no effect of vitamin D supplementation on glucose. This was a small, tightly controlled measurements, no effect whatsoever. We also looked at the disposition index, which is an indicator of capacity to secrete insulin. One of our hypotheses was that vitamin D would work by supporting insulin production in the pancreas. And once again, we saw no difference over this 12-week period in the disposition index resulting from the supplementation. You know, maybe the study was too small, maybe the study was too short, maybe it wasn't the right group. You know, I don't think we can say on the basis of 89 people that vitamin D doesn't prevent diabetes in blacks, but it's not too encouraging to go do a big trial of vitamin D and diabetes in blacks, for me anyway. We had last summer we had a visiting MD from Brazil, and she was interested in vitamin D and inflammation. Of course, inflammation underlies many of the cardiometabolic conditions such as diabetes and cardiovascular disease. And she reviewed the literature, which is very difficult to do because it's just hard to get anything to line up with anything else in vitamin D. So much variability among studies. And I'm not going to go through her tables or anything like that, but just to mention her conclusions, because it kind of relates to what you've seen so far, we already were believing this, but her work only supported it more, that if you're going to study vitamin D, if you want to improve somebody's vitamin Ds, then you want to start with people who have low 25D levels, which just sounds so obvious, and yet you look at these millions of papers that keep coming out and you see people giving supplements to people who aren't deficient in the first place. Not a good idea. And second, that you want to start with people who have high levels of inflammation in the first place, and this would be comparable to starting with people who are at risk for disease. So who are these people? In other words, who is it that we should be focusing our vitamin D work on going forward? Who is it, in this case, who has higher inflammation and lower vitamin D levels? Well, these would be older people because they tend to have higher inflammation, obese people because they have both higher inflammation and lower vitamin D levels, sedentary people, same thing, and of course people having chronic conditions that involve inflammation. Sounds obvious. Okay. Now, I want to show one more paper from another group and this is not even necessarily the key paper, but it's one that really gets across the point that I think is very interesting and a new idea that hasn't been explored very much. So in the previous slide, there was the idea, does vitamin D deficiency cause disease? Well, that's what we've all been trying to figure out forever. And then there's always the possibility in the back of the mind that vitamin D deficiency just is not related to disease. But there's been the possibility raised, and I have not really been able to find out who first sort of published this as an idea, that disease may cause vitamin D deficiency, right? And so this paper, it's only 33 people from the UK. There's no comparison group, but it really gets this point across. They were 72 years old. That they were going in for knee surgery. So before they went in for their surgery, their 25D was around 40, so it was 40 nanomoles per liter, so it was a little on the low side. Within 6 to 12 hours after their knee surgery, look at the decline in their 25-hydroxyvitamin D levels. Well, where did it go? In 6 hours or 12 hours? And, they had some different theories. I'll get to that. But then you can see that over the next 3 months, over the next 5 days, it rebounds somewhat. And by 3 months, it's not quite back to where it was, maybe because they're stuck inside because of their knee surgery. But they had some ideas. One of their going theory they had was that somehow there's rapid uptake of 25-hydroxyvitamin D by inflammatory cells. And we know that many types of cells, including cells involved in inflammation, have both the enzyme for converting vitamin D, 25D to 1,25-dihydroxyvitamin D, the active form, and receptor for the 1,25D, the active form. So these would be autocrine or paracrine actions of vitamin D in inflammation, and I think this is just fascinating. And it's as yet there's no, as far as I'm aware, no really serious attempt to explore this idea in a big population with a clear, set of hypotheses and aims. I mean, it may be going on somewhere. I don't know. So I'm going to end really with questions, because I feel like in all this time, and it's not just me, it's all of us who have been working in the vitamin D field have gone through this sort of cycle of vitamin D cures everything and prevents everything. And now some people are saying vitamin D does nothing. Nobody needs vitamin D supplements. Vitamin D supplements are a waste of time. And I mean, I think, to me anyway, it's pretty obvious that the answer is somewhere in between. Anything involving nutrition is going to matter most in people who are deficient, obviously. But just to kind of think through a few of these questions, I just wrote them out. So how much of there's all this inconsistency that the meta-analysis kind of hide. So how much of this idea that vitamin D does nothing, which is the latest idea, comes from the fact that studies include people who aren't deficient, who aren't at risk for the disease under study? Who should we be studying? And I covered that a little bit earlier. How does the genetic variation among people affect vitamin D requirements? This is a big one. I mean, if I were starting out in vitamin D these days, I would learn as much as I could about genetics. Because there are, we've identified not just the vitamin D receptor differences that I mentioned earlier, but we've identified enzyme genotypes and vitamin D binding protein genotypes that affect either vitamin D levels or their actions that need to be explored further. And that's something that we are trying to do along with others. And if we do find that a lot of the difference in vitamin D requirements depends on people's genetic status, how do we handle that when we develop recommendations, and how do we handle that in clinical practice? And of course, that's going to be true for all kinds of things, not just vitamin D. If we find out that people have individualized requirements, how do we fit that into the RDA way of doing things? And then back to vitamin D binding protein, how do differences in this protein across population groups influence vitamin D metabolism and actions? An area which actually the USDA in our, I think I can say this, in our report has asked us specifically to look at that. They're interested in that apparently. Yeah. What is the time frame over which vitamin D status influences disease risk and mortality? I mean, is it your childhood vitamin D status that matters most? I mean, if so, what good does it do to do a clinical trial for the past 3 years of your postmenopausal life? Clinical trials, they are the gold standard. We think of them as the gold standard, but they can't necessarily do everything. And how does this design, influence the design and the power of the trials that we do? And the slides in the knees, to what extent are low 25D levels the result of disease processes rather than the cause? So I use inflammation as an example. Tuberculosis actually is another example. There's a paper out there that shows that people with tuberculosis have vitamin D levels that plummet with tuberculosis, so it's an infectious process. And do we need to extend our concept of vitamin D supplementation to include restoring vitamin D loss due to disease? So rather than just to prevent disease, if we know people are sick, that we can expect them to be low and we can replace what they have lost due to disease. And finally, what kinds of vitamin D studies should be funded now? I mean, do we want to keep going with these great big giant studies? Do we want little elegant studies? Do we want to go back to basic science and try and figure out how all of this stuff is working? All of this obviously is a huge team effort. It's been such a wonderful experience working with Bess, who's not here today, but she's been a wonderful mentor and colleague. All of the members of all of the teams I've worked with have been wonderful, and I won't go through all the names except to mention Nancy Palermo, who's headed up our studies currently and worked on my diabetes studies and has been a really wonderful colleague. But thanks to all of you, and this is the final word on vitamin D. Am I supposed to have questions? Yeah, Jakob.
Well, we're working the same thing. I mean, what you showed last time, that diseases may cause lower vitamin D. We see this in many, we see, first of all, we see vitamin B6. Inflammation, there's a decrease in vitamin B6. And the main question that that comes out of this. So what happened to other functions of vitamin D when you have a disease that causes lower vitamin D, low vitamin D? The mystery about vitamin B6 is the same as the mystery about vitamin D. And I think you're probably the right track. You know, the question is where is it going? And the same thing about the whole thing about vitamin D. It happens now that vitamin D is on the top. It used to be folic acid one time, and 12. Every time something else changes. And I think your approach of going slowly and ask questions.
Thank you, and I'll read up on vitamin B6. Maybe you guys are ahead of us and I can find something out. Any other questions? Yeah.
So just following Jakob's question. So I was wondering, whether not the vitamin D binding protein really plays a significant role, because all this disease possible they alter the vitamin D binding protein. If you look at all this a study you listed before, they only focus on 25 vitamin D. Not address the vitamin D binding proteins. So you know the vitamin A, if you have a retinol binding protein, they really can prevent the vitamin A degradation. Maybe that similar to the vitamin D or maybe B6.
Yeah, and we are planning to measure that. Or do you want to pick them up?
The question about the drop in vitamin D could it be as a result, as you described before, in the surgery, that there is increased consumption locally in calcium. So in order to get more vitamin coming to transport, and that's caused the reduction.
Yeah, no, that could very well be. Yeah, I think so.
Questions. So if you could comment on the quality and the type of bone in Caucasians versus African Americans, and associated with that, what's the role of body fat mass or BMI, or is it all confounded?
Yeah. Well, African Americans tend to have higher body mass index, and adjusting for that removes some of the difference between blacks and whites, but clearly not all of it. The quality of the bone, there are differences in geometry, for example, hip, femoral neck geometry, and there are differences in, it's been a long time since I've worked on the bone aspects, so I'm not remembering the relative differences in cortical versus trabecular bone, But I will say that the fracture rates in blacks mirror the bone density differences. So whatever kind of the net differences in quality are, the fact that they have bone mineral density maybe wouldn't be enough to think that they were so much better off, but the fact that they have such lower fracture rates is.
So for each vitamin D value, they may have a lower value, but they don't have an increased fracture rate?
We've never studied that. That's a good question. Yeah, I don't know if anyone has. There are some fracture studies, so it might be out there. Yeah, good question.
Related to that black and white difference, I know that environment and seasonality is a broader difference, but has anyone studied behavioral sort of attributes such as the amount of time spent in actually getting that exposure?
Yeah. I did a study. I didn't bring a slide for that one, but I wanted to see how much differences in sun exposure explain differences in vitamin D, and I didn't have African Americans there, but I later did a study. I later helped with a study that one of the students did that saw something similar in black and white adolescents. But in my study, we found that differences in sun exposure really explain only a small difference. And these were among ambulatory people. So, if you compared people who were inside in a nursing home to people who were, running outside every day, I'm sure you would see a bigger effect. But among just regular 65-year-old and up people walking around, we didn't see much of a difference due to sun exposure. It's not that easy to measure sun exposure, but we have,
Is there a difference, or has anyone even measured this, between people who are dark-skinned but not African or African American?
Well, that has been looked at in Polynesians, Indians, some Indian subcontinent groups, and they universally have lower 25-hydroxyvitamin D as well. Whether that's the whole explanation or whether there's genetic differences as well, I don't know. That was done early on, yeah.
Well, the good news, Susan, is you have so many more questions. We'll be hearing you in another 25 years.
Yeah, that's because vitamin D makes you immortal.